Welcome, ladies and gentlemen, to another edition of Medical Hope. My name is Bob Loverin. I'm the host. As I always say at the beginning of the show, we're talking about here is medical advances going on throughout the world and in many, many places, really in every country, really, as advances are happening. And it's amazing, the stuff that's happening. Tonight is a little kind of special note here because as you saw by the title, the coronavirus is in the news all over the place. You hear about it every day, and so many more people have died, and so many more people have gotten it, and people getting on ships, getting off ships, and so on and so forth, airplanes, and being stuck at military bases all over the country, in this country at least. And so it's a very hot topic, I guess that's what you have to say, and so on. But it fits into the show almost exactly, because this is the idea of medical advances. How do you stop these kind of epidemics and people getting killed? The number's now over 1,000 people killed, although most of those are in China, which is not good either, of course. At any rate, we have with us this evening a young lady, a PhD, Dr. Judy Mikovits, who's been on many shows before. And when I got this, thinking about this coronavirus, I thought Judy is a perfect example of somebody, a perfect expert, I would have to say, expertise at least in this area. And so I called her to do a show, and she's with me this evening. Judy, I always like to shake hands to let you know, even though I know you very well, obviously. Right. I've been on many times. Judy's a very good friend of mine, and a very passionate person about these about things like that we're going to talk about this evening. And I'm going to put a little different spin, I think, on this whole idea of viruses and the susceptibility to people for it, and vaccines, basically. Anyway, without further ado, I think, Judy, let's just begin the show. Give me your first slide and let me introduce yourself.
Okay, well, thanks, Bob. As you say, I have particular expertise in this because I've studied not only RNA viruses, retroviruses, and their effect on the immune system, and thinking about how emerging coronaviruses, what they mean. I simply went back, when you asked me, to some reviews from 2016. So I show here, we recall in 2002 we had SARS, which is the severe acute respiratory syndrome coronavirus. And again, that one...
Stop for 1 minute, though. The coronavirus is actually a family of viruses.
Correct.
SARS being one of them.
Correct.
I guess MER, we'll get into that.
MERS is another one.
Of course, the current one.
Right.
They've named it, in fact, they said it was a coronavirus, and all of a sudden the news just started talking about an NCOV. The N stands for new.
Coronavirus. Yeah, so they're labeled. So SARS was named SARS because it causes a severe acute respiratory syndrome, as in fact most of the coronaviruses cause respiratory cough and hemorrhaging in the respiratory tract, in the nasals. They call it nosocomial.
Yeah, you can't really see this very well, ladies and gentlemen.
Nosocomial outbreaks.
Anyway,
so this is a 2016 review, and it just shows you that family of viruses and what the structure of the RNA virus looks like. So we know a great deal about these viruses and their interaction with the immune system. So we appreciate their receptors, how they infect. So the SARS coronavirus, as you mentioned, and the MERS coronavirus, how they enter the cell through the receptor. And in the case of the new coronavirus, they enter through an angiotensin receptor. So, and in fact, it's very interesting because the downregulation of that receptor actually saves you from damage, because when those receptors are upregulated, they cause microvasculature leaks. So you start bleeding and hemorrhaging, and that's in fact how these cause death. But so we know the biology quite well, and I'm not going to go into it in great detail.
This is for all of the different coronaviruses.
For all of the different coronaviruses. They have different receptors, and they have different capsids on, so they have different points that bind with that receptor, and they go into the cell, enter, and they uncoat in the endosome, and then they release their genomic RNA, and they have an enzyme which replicates that RNA, and it goes to the nucleus of the cell, and it makes a virus particle that looks like that, represented schematically.
So I'll go back to that slide a minute.
Sure.
Just so people understand, this is what's called, this is the virus itself,
Yeah.
particle, and it's very, very small.
Correct. Very, very small.
This is the size of this compared to a bacteria, which you can't see in a microscope either. This is 100 times smaller than the bacteria.
Correct. Somewhere in the nanomolar, 10 to 50 nanomolar range would be this size of a coronavirus.
And these little points along the edge here are the receptors.
They're what tap into the cell.
That tapped into the cell. But that has to match up with the cell itself.
Yeah, it has to see the receptor on the cell.
It doesn't go to anywhere. It goes only to the receptor that it's this.
Correct.
It's like a puzzle being fit. This is the piece that fits into that part of it, basically.
Correct.
Okay.
And and then once it gets into a cell that it can infect, that has receptors, so lock and key kind of idea.
Yeah.
Once it does that and it latches onto the cell, it opens up, goes into the cell, uncoats, and makes many, many, many more particles using your cell's machinery.
You can't blame the virus in a way. It's trying to live. It can't live without, it can't multiply basically without going into another cell. In the process, of course, kills that cell.
Correct.
Or does damage to that cell.
Does
or even
damage to
cancer, in fact.
Correct, and that's a long-term thing, but you're exactly right. A virus can't kill its host. So that's a very efficient virus, and so that's why we're talking about, and we'll talk about it a lot more, about how it evades the immune system. So it's the innate immune system that provides a strong antiviral response. So the type 1 interferons, that, remember, all the way back in the 80s, that was my first job, the magic bullet against cancer, purifying interferon and using that as an anti-tumor, anti-cancer agent. So we now know, 40 years later, we know much more about the immune system and the various parts of the type 1 interferon pathway that these viruses evade, and so they're able to suppress the type 1 interferon response, and that makes many, many, many more particles, and that stimulates a very strong inflammatory response through NF-kappa B and the cytokines that we've talked about on this show endlessly, IL-1, IL-6, IL-8, and so this is a very powerful, inflammatory response, because the host, the immune system, sees that as, uh-oh, we're in trouble, and in fact, it's that severity of the immune response that does a lot of the tissue damage. So if you can, the virus has parts proteins that suppress the type 1 interferon, so you can continue making more and more. Then they infect more and more cells in the host, in the lungs, in the nasal passages, and that's when that strong inflammatory response, you turn up the fire, and it's actually the damage done by your own immune system that kills you.
You're damaging the virus's cells then, right?
No, you're damaging, you're killing the tissues that are infected. So first you want type 1 interferons to suppress the replication. Don't make many more particles. So the virus actually makes proteins to tell interferon right here don't go, leave us alone, we're fine, you don't have to worry about us. So they suppress the type 1 interferons. That allows a lot more particles to be made. This last picture, that's a particle. So then you've got a lot more particles, and you've got a little virus factory making particles, and then the innate immune response in your cytokines and chemokines, these particles are recognized by things called pathogen associated molecular patterns. So it just sees a pattern and it stimulates, it turns on the fire. It literally says, we need a fire engine, go to the fire and put it out. And when the immune system is going there with all these cytokines and chemokines, it does a lot of damage to the tissue. And
now
so
you say to the tissue of what?
Of your lungs.
Of your cells, right.
The humans.
But that's good.
It's not good. It's
not
how
good.
it
Okay.
kills you.
Yeah.
So
Okay.
what you've got to do in order to treat
suppressed.
or prevent is you've got to lessen all the inflammation so that you can really focus an immune response on that.
Yeah.
And so we can actually think about why the bats, and I believe I have that on the next slide. Well,
Yeah.
this talks about the emergence.
Yeah.
So this is what we're told, and this is one of the frustrations. So we were told with this virus outbreak in late December when we first learned of it,
Yeah.
that it came from a fish market and it just emerged from a contaminated piece of food.
Yeah.
And one woman got it and then it spread around. Well, what we now know is that there's a biosafety level 4 laboratory where they were manipulating. It was because of SARS and MERS and the severity of these that we had been working with, and I say we as scientists, both in the United States and in China, in our biosafety level 4 labs. I worked at the one at Fort Detrick, where the National Cancer Institute. I worked in the early 90s with Ebola, trying to teach it to infect human cells without killing them from that immune response. As you said, the cell will blow up and it'll kill, and a virus doesn't want to kill its host. So this virus was actually the SARS viruses. The bats were taken to the Chinese facility, a biosafety level 4 facility in the Wuhan laboratories.
Right.
So that fish market was actually very close by, but
within a few hundred feet.
and so it's very clear now that whether through safety measures or whatever weren't taken, but the virus is a more pathogenic virus. So they didn't in fact attenuate the virus, which they had made by passing it through and trying to do what I do, teach it to infect human cells without killing them.
Yeah.
The attempt, if you just say what we were doing in the lab, the attempt was to weaken it so that we could have study it and develop effective treatments for it back in the 90s when I worked with Ebola in the biosafety level 4 at USAMRIID. But what happened is, some of the virus escaped, and the bats have different immune system, and we'll talk a lot about that because it's interesting. But what happened here is like from 2002, so this is greater than 30 years ago, and some camels got infected over in India, and so that's the Middle Eastern respiratory syndrome. That's what the RS. So there's SARS, severe acute, because this was an acute infection that would kill them. And it's very clear that it doesn't go through a secondary host. So the bats can interact with other small animals and small mammals, and then humans come in contact as suggested. It cross species. It's called zoonotic transmission, zoonosis. That's how a pathogen jumps
Zoonosis. Define that.
So the zoo is an animal.
Right.
So you transmit from one animal to another.
To this animal
species.
here.
Correct.
With the humans,
Correct.
of course.
And usually the doctors get infected, the scientists get infected. Me working in the laboratory, I'm at risk of getting infected, so that's why I was working in a biosafety level 4.
Right.
None of my air came in from the lab, and none of the lab air escaped. But of course, somehow this virus, this now very highly pathogenic virus, that doesn't need a secondary host, that doesn't need to go through a secondary host, can actually go directly from the Wuhan facility into men, and through the doctors, through the scientists, and then through community contact. And then when the important thing about the patient in the hospital is they're already sick. So they have a compromised immune system, so they already have inflammation going on.
That's true.
And they're already susceptible. And if you have another pathogen directing your immune system away, then it allows another pathogen to come in there and wreak havoc because your immune system's already compromised. And so we could talk about this fascinating subject.
Well, certainly people in a hospital are already sick. So
correct. And so
they're susceptible to it, plus older people and small children, too.
Yeah, well, the very elderly and the babies, so they don't have developed immune systems. So they don't have that type 1 interferon pathway, and often it's compromised in sick people. So you're going to spread it more through the hospital.
So this is kind of what's happening. What you're telling us here is that's what's happening. How it got started now. This corona NCOV. That's how it got started. It was already created by this the
laboratory manipulation,
and because they weren't as careful as they should have been, which is tough to do to begin with. It was bound to happen, I guess. That's what, in a way, it's a sense. It's bound to happen.
It's happened multiple times in the 21st century. The first one was 2002, but we know, and we'll talk about it a little bit more, but we know the issues with the XMRVs, the mouse viruses that contaminated the blood supply, and probably came from the polio vaccine all the way back in the 1930s, because the polio virus was passed through mouse tissue, mouse brains, to attenuate it. So picking up dormant tissues, and then 60 years later, we isolated infectious and transmissible viruses that from people with diseases like autism, ME/CFS, lots of cancer, and this was the big problem of 2009 in our discovery. But again, there was an outbreak of Ebola in 2014, and it was very clear. It was said that virus came from somebody not cooking their food properly over in Liberia. That was 21,000 people died in Liberia from the Ebola outbreak of 2014, and it was very clear that the strain of Ebola wasn't a natural strain that passed over 30-some years the way we're talking about. It was actually accelerated likely by the manipulation of the very work we did in Fort Detrick, because it was very clear that the strain of virus had different mutations that enabled it to infect the cells, and spread more quickly.
Playing God.
Playing God.
And you're not.
And we're not. And so, So what I show here, that when we think about coronaviruses and why the bat viruses are so deadly, what's pretty cool about this is
keep in mind too. Get this this idea across. That bat can fight this virus. The bats don't die of the virus. They can pass it on.
Exactly.
They don't. Obviously, they're still going to be sick or something like that. But this virus, they've developed an immune response to this virus that works okay. So when the virus, but they still can pass it on. Unfortunately, humans don't have that immunity.
Type of immunity.
Right.
And that was exactly what we were talking about. So what the bats are the only mammals that fly, and it takes a tremendous amount of energy to fly, and that produces a lot of reactive oxygen species. We think about when our athletes and all the damage from reactive oxygen species and using anti-inflammatories for pain and things like that. So reactive oxygen species are bad.
What is that reactive oxygen species?
It's like I'm trying to think of the word to use here. Free radicals. So free radicals damage tissue. They damage mitochondria. It slows you down. It's it's called free radical damage, and we think of that, and we have that's why we use all these antioxidants. Blueberries, strawberries, all our colored fruits have antioxidants and dampen free radicals. So with the bat, in order to produce that much physical energy, has a very effective way to keep those free radicals, because they'll damage your tissue. They, you know, the free radicals are oxygens that are like O3, and it does damage to your tissues. So if the bats remove that very quickly with anti-inflammatory pathways, they turn off that fire I was talking about, the IL-1, the IL-6, they turn it off very quickly, and so that they don't die. So they don't accumulate the tissue damage, so the virus lives quite comfortably with them, because they're not doing damage through their own immune response, because they have a very efficient system for clearing out free radicals which cause tissue damage from these things. And most importantly, they have a hair trigger release of that signaling molecule I just talked about, interferon alpha. So that's an antiviral. It's the most powerful. I brought a bottle of it with us. You know, that was my first research job at Fort Detrick. It was purify interferon alpha and give it to humans. It was the magic bullet of cancer, March 31st, 1980. That was just before I graduated from University of Virginia, and I said, that's what I want to do. So I got a job June 10th, 1980, started put the first interferon that went in for cancer.
The magic bullet.
So the magic bullet for cancer.
And it turns out it was.
It turns out it was a very, but we don't use it anymore because we didn't think about it. So I brought this is a homeopathic called Paximune that some colleagues have made over the years, and you can buy this on the internet, and I have it in the slide, because you could spray this in your nasal passages in your lungs.
Spray, right.
And so you would have your hair trigger antiviral right there. So you need nothing other than this. Vitamin C will dampen down that NF-kappa B inflammatory pathway.
Right.
So we have things ready to go for these viruses. In addition, should you be exposed to keeping a healthy
are natural
immune system,
products now.
which are natural products,
Not
and
synthetic,
so they're
not making up stuff.
not synthetic, not recombinant, not gene expression, and so. Interestingly, so this I show here, this Australian black flying fox.
You can barely see it. There's a bat right there.
There's a bat right there.
Hanging upside down.
I never did like bats.
Yeah.
and so sometimes this virus, which also can cause severe respiratory disease, is transmitted to horses and sometimes humans. Well, interestingly, one of the old hormone therapies I used to work with, again, I did drug development, was called Premarin, and it came from pregnant mare's urine. So you would use an anti-hormonal therapy. So we would have to make sure in my job, and I have through the years, that none of these viruses were present, so that the process of making the drug would kill the virus, which heat will kill the virus and other things will kill the virus, and simple interferons. So there are ways that we can use, have our knowledge of these viruses and keep them from emerging. But what's really become clear is that it's the manipulation of animal tissue in humans, and how we can be accelerating the this circulating. This is another cluster rate. So here's the host range, and you get recombination events if you have more than one virus.
What do you mean by recombination?
So if this takes on a mutation, and it increases the host range. Correct. That's a normal cell, and there's a virus in there, and it takes on a mutation so that it can live longer. And then
that's to keep in mind. These viruses are constantly mutating.
Correct, constantly mutating, and there's error in their enzymes, so they can recombine with other viruses and make new viruses. And this, of course, is what's happened in the case of the XMRVs, the recombination events that allowed new infectious transmissible viruses to be generated in only 2 weeks in culture in one of our laboratories. So we accelerate that 30-year time frame that I showed back here. This is cross-species transmission, takes greater than or equal to 30 years. Not if you're running it through Fort Detrick or
we'll go back to that slide. They keep in mind, the camels, this was passed on to camels then at some point?
Yeah.
Somehow or other?
At some point, yeah.
That's what MERS stands for, basically.
Correct.
It's for this one here. I'm not sure what that means. It's, what does that stand for? It's
MERS, Middle East respiratory.
Middle Eastern strain and so on. This is the
respiratory syndrome.
Now this is the Liberian thing, or that was Ebola.
That was Ebola. Was Liberia. So it's and again Liberian. Similar family of viruses.
Yeah.
So. But yeah, I mean, we use camels for food and for fat, and we actually use them for some of our products and therapies now because the hump fat itself contains a great deal of nutrients. So again, all you'd have to do is make sure you heat it, you decontaminate, you autoclave these things so that they're not transmitted.
But you have to make sure of that.
But you have to make sure of that. So cooking and things like that. So here you end up with these direct infections, and whether you go through a secondary host or not. See, when you're in the laboratory and you've taken on a lot of mutations, that's what I was talking about with the Ebola in Liberia in 2014, a lot of mutations. So it was easy to get an epidemic strain out. And then of course, one of the things that we do that is not correct.
Now, when you say epidemic strain, what's that mean?
That means one that can transmit much faster and infect a lot of people and cause severe pathogenesis.
Right. It's a strain that's going to cause it, and it's quick.
Most of the coronaviruses and other RNA viruses are harmless.
Now, keep in mind also that this is it also indicates, talks about, it goes back to what a virus is. One of the differences is different shapes for one thing, but RNA. Some of the viruses are just RNA in it. In other words, and the other ones, some of the other ones are DNA.
Correct.
When you're talking about the RNA stuff.
Viruses. The RNA viruses are a lot more dangerous than the DNA viruses.
The RNA are a lot more dangerous because they can do combinations, whereas DNA has to be, certain things have to go together.
They're a lot bigger. So the DNA viruses are the herpes viruses. And it's interesting because the type 1 interferon pathway doesn't control, isn't the front line of defense for the herpes viruses. There are other pathogen-associated molecular patterns and pathways to heal those. But again, if you get a co-infection with both, you can create a much sicker patient. The host becomes much sicker because its immune system is further compromised and can't respond. I can tell you a good story about Liberia if we have time in the end. But so it's pretty clear that this coronavirus didn't originate in a fish market or in a market that
it wasn't
came
natural.
From the lab.
It wasn't natural. It wasn't natural. It was made in this laboratory. Doing research, I guess, or something like that. Although it's been associated with biotech warfare and things like that, that would make it, at least they knew what they were doing. This in a way they didn't know what they were doing.
I don't think we know what we're doing. I think we knew the risks of what we were doing. I think it's clear that we've known the risks now for at least 50 years of mixing animal and human tissue.
And when we say we, it isn't just the Chinese.
That's the royal we of the scientists in general. You know, again, I was one of those scientists once doing that, thinking you were doing good because you would learn how to develop drugs against it. But as you see, we have learned how to develop drugs against it.
The bat escapes from the cage or something like that.
Yeah, with the type 1 interferons, and we developed them a long time ago.
Doesn't close the door or something, and next thing you know, you got a epidemic going on.
Exactly.
A real big problem.
But again, most of them, it takes quite a while to acquire the ability to infect easily human cells. And that's how we can tell this virus
now, you're saying but that's through mutations then?
Correct, and that's why we can tell this virus didn't come from a natural setting because of the kind of the ACE receptor, and that's a lot more detailed than we need to go into for this show. But anyway, this is
but the bottom, it has to adapt. I mean,
correct.
It's because it's not going to pass along. Because it's mutating constantly and so on, it's finding a way to do it, finding a way to stay alive. You may have a lot of these viruses that can't do it, but if one of them does, that's all you need is one. And at that point, it becomes many.
And when we're working with cell lines in the laboratory, they're continuous cell lines, meaning they grow all the time. That doesn't happen in nature. So we're giving it a huge acceleration of the zoonosis in laboratory
which
manipulation.
even works. Whereas it takes several million years and something like that, we're doing it in a day.
Or a few weeks.
Few weeks or whatever.
So this particular slide, and I show this because again we're going to go back to vaccines, because when you make a vaccine, you grow a pathogen, an RNA virus or whatever it is, in a tissue, in either an animal cell line, fly, mosquito, human. Chickens, flu vaccine, and we'll talk about that. So I show here particularly Pediarix.
Now, these are two vaccines essentially.
Correct.
That are given to 2-month-old children.
2-month-old infants.
Infants and so on.
2 months, 4 months, 6 months.
And these little letters here all are different diseases. It's a combination.
Yeah, vaccine.
A vaccine
so this
given
is
to a 2-month-old baby.
Correct.
Who doesn't have an immune system.
Correct. And it's really
Very
important
little, at least.
as we look at this in the context of an emerging zoonosis and emerging viruses. So this is diphtheria, tetanus, acellular pertussis, Hep B, and polio in a single vaccine.
Yeah.
But what's more important is I'm showing here what the CDC table called the excipient list. This is what else is in the needle.
Yeah.
And so in order to grow, each of these antigens has a different host that it grows in. So in order to grow these different antigens and make a vaccine by mixing them all together,
are they making it just antigens? The antigens are what antibodies attach to on the cell. Okay?
Well, so a weakened virus, this polio virus, that's the big problem in this particular one that for today's discussion. That polio virus is a live attenuated virus. And so in order to grow that polio virus, which is an RNA virus, we grow that in Vero monkey kidney cells. What is an intermediate host for coronaviruses? Monkeys.
Yeah, right.
The Ebola. And so Vero monkey kidney cells, we grow viruses very well. You know why? They have no type 1 interferon alpha. None. No. So you're a little virus replication factory.
Right.
But the problem is they don't clean these out of the vaccines.
I didn't realize that interferon was that important.
Very important. Type 1 interferon, my first job. So at any rate, they don't clean it out of the vaccines. So now this child, should this child be exposed to somebody who has a coronavirus, they've got the makings of the coronavirus right there, and Ebola and HIV, because they have the genetics and the viruses that were in Vero monkey kidney cells injected into this child, and you're a factory for something else, and you've stimulated a very strong immune response with the aluminum hydroxide and the aluminum phosphate. I mean, these children, especially vaccinated children, if exposed to this coronavirus, wouldn't survive. The same thing you could say, and I didn't put the excipient, I might have it here for the flu vaccine.
Whereas they might very well, it would just be without this thing, they might very well have had the ability to fight off this.
Correct.
Correct.
They might be, and so would the mothers. And so, unfortunately, the mothers are being vaccinated as well these days, and that never happened when I was a child or when I was a childbearing age.
I got measles. I didn't have a vaccination.
Correct.
I lived through it. Which,
Correct.
by the way, keep in mind too. We should add this to it immediately here too. These vaccines prevent this disease this time around. But unlike the immune system, this is only the the innate response. So but that doesn't develop an immunity to it.
A lifelong immunity.
The adaptive response.
To any of these, and you can even argue, because there's no research that's ever been done in a 2-month-old, that they don't develop any kind of immunity.
Yeah.
So these given the way they're given in the 21st century, they're all risk and no benefit.
Yeah.
And all risk. So,
but the benefit has to be beyond you know, beyond a couple of years, well, a couple of months even, and so on. Otherwise, you're vaccinating them continuously, basically.
Right.
And that certainly isn't good.
Well, and this, of course, is the zoonosis of the retrovirus. This was our work in XMRVs. This was our 2009 science paper that was, you know, basically ended my career, because when the magnitude of this zoonosis, that mouse leukemia, mouse cancer-causing viruses, had been transmitted for now more than 50 years. I think the first outbreak of myalgic encephalomyelitis and the first cases of autism were a zoonosis that came from a polio vaccine. And remember, in the 30s, in Los Angeles, in the 30s, we vaccinated, we attenuated polio by passing it through mouse brains.
Right.
So we picked up a mouse retrovirus family that is associated with many diseases: cancer, chronic fatigue syndrome, autism. Passed it through the doctors. The children got it. The whole family got it. And so the susceptibility here was this gene called RNA cell. So it's like a Pac-Man RNA cell, ACE. It clips. It cleaves all RNAs that are outside the cell. Okay. RNA viruses, DNAs. So that's a simplistic way to say it. So this mutation that was found in prostate cancer susceptible people, so only people with prostate cancer that had this R462 mutation, one mutation gave the enzyme about half its ability to degrade these viruses. So those men were more likely to get inflammatory and deadly prostate cancer very young. And so then that was where the virus was first identified in 2004, the sequences, and then we found it. And of course, as I mentioned in the slide before, there's cow blood in every vaccine. There are pig retroviruses in the Rototec. There's vero monkey kidney cells, which carry SIV, which is the progenitor of HIV. And then there's human fetal tissue that contain a lot of endogenous viruses from humans.
So what you're saying basically is the polio vaccine and other vaccines, but polio was kind of, I don't know, pretty much one of the original ones at least. There's been documented cases where people have gotten sick, cancer. I mean, keep in mind that for people that don't know, viruses can become cancerous.
Correct.
They just don't make you sick. They give you cancer.
Right, and SV40 is another of the viruses that causes lung cancer, associated with lung cancer and other things. So these zoonoses are not new, and we know of them, and they can be critical because, of course, retroviruses aren't easily cleared, and they take 20 to 30 years to kill the host.
Right,
but
we don't
as
know. They're giving this thing to two-month-old babies.
Correct.
You may find it 30 years from now.
Well, and that's exactly what we're seeing.
We're seeing right now.
Cancers. We're seeing 20-year-olds with breast cancer, triple negative, one of the most aggressive cancers. So this is just what I just said. So how do mouse retroviruses get into human? Well, you take the mice and the conveyor cell.
What is a retrovirus then?
HIV. They have both RNA and DNA. And they reverse transcribe their RNA into DNA and they insert themselves into your genome for your life. So if they hit a stem cell, if they hit a blood cell, an immune cell, you become a little virus factory, and this is what it shows. And then we're taking them we're mixing them. So I use the blue and the red here. So we just take the mouse and we mix it with those other cell lines. The cow, the pig, the calf serum, the vero monkey kidney cells, the MRC5, the human cells. And then we take the whole gamish. We don't even clean it up. And we just inject it into a vulnerable infant. And I show a black baby here because that's what we learned in the vax situation with MMR. MMR are 3 RNA viruses. And so if you have that, blacks and Mexicans and certain other people have more of that R462Q, where they can't degrade it as fast. So what we learned is in their enzyme system, their immunity isn't developed until they're 3 years old. So what we learned in the data that was covered up by Thompson and the CDC back in 2000 is if they waited until a black child was 3 years old to give MMR. They dropped the relative risk of getting autism by 4-fold. And of course, they didn't wait. They just spread it around.
And just because they are a different ethnic background, or what do you want to call
Different
it, and so on.
gene
Different
susceptibilities.
genes. They have
Correct.
more susceptibility. So, but they got the same vaccine.
Correct.
Which had been tested on white kids, I guess, and so on.
It wasn't tested on anyone, actually.
It was tested on anyone, okay.
So everybody had more of a risk because the contaminants haven't been advertised.
Yeah.
So I just show a simple slide of what I call 21st century acquired AIDS, acquired endocannabinoid, because the endocannabinoid system, now we know we have an endogenous system of ion channels and channels which can send signals to the immune system and calm everything down, and that's cannabis as therapy. So I'm proposing for coronaviruses, for emerging viruses, for zoonosis, to prevent tissue injury, we can use cannabis if we use it in a rational way.
Be tested,
Absolutely.
When you say cannabis, because everybody thinks of marijuana and so on, but that's not the same thing at all. This is cannabis, which is very natural again. Keep that
from
in mind.
plant,
Very natural in nature.
We're not talking heating up smoking. We're talking about taking a plant in nature and taking cannabinoids that we know will calm down that fire. So the endocannabinoid system in simplistic terms is a system whereby it's a dimmer switch on that inflammation. So in the beginning, when I said if the fire gets too high in humans because they can't clear the reactive oxygen species fast enough, it doesn't do that in bats. Well, the endocannabinoid system is a system where with a plant you can put the dimmer switch on it. You can not the on and off switch, the dimmer switch. Just stop the flames of the fire, the inflammation. Stop the flames from going that high. And so, that's. And I show here another slide of the 2014 Ebola outbreak. And so I mentioned this, but this is what came out in the press about in Liberia. And these were our NIH directors. And when you inject a vaccine and bypass things or do anything where you mix blood, because of course in HIV/AIDS it was the blood supply that was heavily contaminated and spread that through the susceptible populations. And interestingly enough, until very recently, we didn't vaccinate AIDS patients because we knew we would kill them. We would spread the virus through their body, where we could shut it down if we allowed the natural immunity to work, which was of course some of my PhD thesis in the role of the macrophage in controlling the disease. So in 2014, I show here this was from, I believe it was an article in
Scientific Reports?
Scientific American or something like this. So here's what the CDC said when it was very clear that the Ebola in Liberia didn't spread through blood and body tissues, that it had been aerosolized, that it could be spread the same way as the emerging coronaviruses we're talking about right now. So the CDC says, well, it's not contagious because when you cough with measles, it goes 6 feet. I'm not sure how they counted the drops. And if you cough with Ebola, it only goes 3 feet. Therefore, you don't have to worry about it, folks. So this is the kind of media, and we're seeing it in our media today, the lies that are being told by our governments and our labs about these outbreaks and about the origin.
Well, that makes not so much lies that they don't understand it themselves.
They do. I mean, they do. I think my two books will tell you, well, here's Harold Varmus, and he was the NIH director, and he implemented the xenotransplantation program in 1999. We remember when we used to use pig aortas and veins for heart transplants. We didn't put those in HIV-infected people because of the risk xenotransplantation. You take foreign tissue, xenotransplantation.
Right.
Take foreign tissue, xeno, and put it in another species. And then that runs the risk of transmitting or accelerating a zoonosis because, of course, a patient with HIV is compromised because the HIV is compromised the immune system. So they said in some cases, the viruses that are not pathogenic in their natural hosts can in some cases become pathogenic when transmitted to a new species. So they said this in 1999. And we knew this. We've known this for most of my entire adult career. And we were talking about your friend with Lyme disease.
Yeah.
So it's no different than the multi-component animal viruses from mosquitoes. So what accompanied the Zika outbreak and the emergence in Brazil, which according to the news, caused the microencephaly, the people born with deformed brains and small brains and from pregnant women. And so that the Zika was, supposedly passed through, mosquitoes, but or animals are just somehow zoonosed into humans. But at that time, what they were doing in Brazil was they were spraying the natural environments, and they released some engineered mosquitoes to the area, and then did spraying with a lot of pesticides, and then they vaccinated with DTP.
They were testing this thing on mosquitoes, but unfortunately, some of the mosquitoes escaped. Is that what you're saying?
Well, no, they released them on purpose,
okay.
and
well,
as a way to control, theoretically, other again, playing God as a way to control the mosquito population or pathogenic mosquitoes. But what in fact they did was they caused the zoonosis of the Zika, which is also an RNA virus.
Because it mutated? To the point?
No, because the animals under stress, when even bats in the rainforest there, they took them out of their natural environment and put them under stress by all the spraying, and then they released mosquitoes that were engineered to produce other things. And this paper is a 2016 paper which shows they could get all kinds of viruses out of those mosquitoes. And so you think about, and again, an expanded host range to include monkeys. So now here's a multi-component animal virus. And again, this is published 5 years ago. So when you say they don't know, and nobody sees this in the news, nobody sees this fanfare, and we think about Lyme, which is Borrelia, ticks. And ticks have been weaponized and released. We've always had ticks in our environment. Why do we have this explosion of tick-borne diseases?
Right.
Well,
Lyme
it's the
disease.
crippling, and because there are other pathogens emerging that are crippling other halves of the site. So so there's and these are being caused by our manipulation of animal tissue. And then this one shows you, this is the MMR5. So this is a recent MMR vaccine, and an independent group over in Italy got a hold of some because, again, these vaccines have never been tested. We don't know what's in them. Nobody's an expert because we don't know what's in them. We can't even guess. But this group got some vaccines.
They don't know what's in them.
Because they're not tested.
What they think is in them,
Yeah.
but it turns out to be.
Not what's in them.
There's other things in it.
Right, and they won't test them, and they haven't been safety tested, as we know, and that was actually admitted by the WHO and the CDC that these vaccines are killing people, and that there's a lot of contaminants. And yes, they've never been safety tested as required by law since 1986. Not a single test. So here's an independent study that shows, 32 sequences of a human endogenous retrovirus that's associated with a lot of diseases like Lou Gehrig's disease, again, explosion of these diseases. Equine, this is a horse anemia virus. 2 of those, avian leukosis virus, It destroys your white blood cells from birds, and 32 4 sequences of
So
a
what you're
different
saying, these
herb.
viruses were in that.
These were all in MMR.
And they didn't. Oh,
And that,
I guess it's worse. They really didn't know it. They didn't test it, so they didn't really know what was in it.
well, but it's mandated by law to be injected in our children or they don't go to school.
Yeah.
So this is our problem.
Yeah.
So it's all right if you want to take this stuff, but we don't have informed consent.
Yeah.
We're being lied to about what's in these, and then
Yeah.
it's being mandated by law or our
Well,
ability
it's not so much
to
that
have a
they
job.
lied to. So they didn't know what's in there.
Well, they won't test it.
These children are becoming guinea pigs.
Yeah, and our adults.
Yeah.
With every flu vaccine,
Well, they become
they're not
adults, and
tested.
then like I say, 30 years from now, you really don't know what
Right.
caused this to happen. Well,
Right.
it's because you got a
Right.
vaccination of something that really weren't
Right.
sure about.
And
they had
that's-
never tested it.
That's what we're seeing, and you know, as required by law.
Yeah.
I mean, I tested this
Okay.
for years. So
Okay, we only got a
that's,
few minutes left here, so
Yeah. well, and this just shows you the antiviral pathway of the cannabinoids, so that we can start thinking they're natural product medicines.
Mm-hmm.
So the cannabinoids here's type 1 interferon. So they block that inflammatory tissue damaging response.
Mm-hmm.
They'll control the fire, as I mentioned, and they'll stimulate the production of type 1 interferons. So you can take a cannabinoid formulation from hemp, as you just said, non-psychoactive, and you can dampen the pathology that would kill you, and spray some type 1 interferons in a homeopathic form,
and
like
here
I
again, we're
have
talking
here.
about a natural product here.
Yeah, and a natural product that will prevent and treat the coronavirus emergence.
Yeah.
And in fact, any of the other viruses I've been talking about tonight, including retroviruses, even if exposed, we can prevent disease if we allow. These are again illegal. We can't grow, use, make drugs out of these plants, and they're just like any other family of plants. That I've made drugs from for
it's
now
got a bad
40 years.
reputation.
It's got
I guess
bad
it's not a good
reputation.
reputation among a certain group of people and so on, but
Yeah.
the bottom line, this is not marijuana here.
Right. It's time to end the plague of corruption
Yeah.
in what we're allowed to use as medicine,
Yeah.
and what medicines are being forced on us, which is basically
Yeah.
all that we're trying to say. But so the emergence of the new coronavirus, the N coronavirus 2019, is not unanticipated from what we're doing,
And very appropriately, you've written 2 books. The latest one, in fact, you want to show that here?
lots of plants.
Or do you have it, Fred? No, I guess not. Okay, well, here are the same.
Yeah.
These are the 2 books. This has both of them and so on. So if you're interested in finding out more about them, this one has not been published yet, I gather, right?
Yeah, this will come out in about a month.
Yeah.
What's wonderful about this book is, so the first book, as you know, we talked about the censorship and the cover-up of the XMRVs,
Right.
and the idea of the diseases
Yeah,
they were
which you were
causing.
very much involved in.
And I only learned the hard way.
Yeah.
But in the plague of corruption, it starts really from 2014, stuff that I didn't know that we've even talked about tonight. And what I said is, in plague of corruption, we raised the broader question of the enormous risk of using animal tissue in research and mixing it with human tissue for the development of medical therapies, while covering up the value and the efficacy of natural product therapies like cannabis, homeopathy, energetics therapies like we've talked on this show we've talked about electroporation. And so the energetic therapies are healing as well, including even light therapy. We can restore and reverse some of the Alzheimer's effects with different light energies, different therapies with light. So the plague of corruption, that's why we titled it this way, and Frank coined the phrase restoring faith in the promise of science. As you know from this show, and why you do it is medical hope. We have huge promise for science. We have the technologies to do these things. We can catch these outbreaks very quickly, and if we don't censor the truth when an unfortunate, inconvenient truth comes out, like a practice that we thought might be risky, and we ignored the risk and allowed the emergence of new strains of
I got a couple
viruses.
minutes left here. One thought that there's a paper out today, I guess it is, from the University of Texas, Austin. Here's a tool, you're talking about the science, a tool that they've been able to sequence the genome of this new coronavirus, this N, CoV. But what they're trying to, the last paragraph of their paper, at least the one part that I read and so on, they're taking people who have basically survived this. There's only, 2% now, only, but 2% of the people who will get it end up dying, and that's, of course, old people and babies and people who have a compromised immune system to begin with. But because the fact that these people can do and again, here it is, they get the same, they get the innate response, but they have the adaptive response, the antibodies, which means antibodies, basically. Antibodies, little soldiers running around your body and so on, looking for this same virus. If it shows up again, they got it, right?
Right.
Getting those antibodies out of people and so on, and then reproducing those to help people out and so on. But here again, it's a natural thing.
Yeah, appreciating the memory responses.
Right.
We can go a long way towards healing.
Because I always tell people it's taken millions of years to perfect, and God, evolution, nature, whatever you want to call it, it worked pretty well for people, basically, as a system, and we're trying to do it in a couple of years.
Right.
We're playing God, basically.
Right.
Anyway, okay.
Some of us have a problem with that.
Yeah, some have a little problem with that.
Right.
Well, I hope you've understood some of this stuff, and it gets a little better, a little different slant on this whole virus thing. I think that we'll get over it, basically. It'll pass. It always does. Even in the natural world, it always passes, because your immune system is adapting too. Anyway, thank you very much, Judy, and I hope you've learned something. We'll see you again.
Thank you, Bob.